Good afternoon. Welcome back to Zoology second year class. We are in the SAQs already. In the evolution topic, we are there in the SAQs. Next, in the genetics, two questions are left. That answers we are going to discuss. Next here. Here in the genetics, the four questions are important questions are there. They are first one, sex determination in human. And second one is the erythroblastosis fetalis. Third one is the genetic balance theory. Next one is the HGP, Human Genome Project. Already we discussed about this one. The first question we completed and the second also. Next, the third one, what it is means the genetic balance theory. Here, genetic balance theory of the determination. So this mechanism is determining of the sex. That is mainly the observed, studied by the Bridges. Here, he observed in the Drosophila. Next, it is found in Drosophila. Y chromosome is not necessary for the determination. In this, according to the genetic balance theory, what it is means in the Drosophila, Y chromosome is not participate in the determination of sex. He explained about this one, and here it is normally, for example, Y chromosome is there that we considered as a maleness, and the X chromosome is there that is called as a femaleness. But here Y chromosome doesn't play any role in the determination of the sex. That is called as the genetic balance theory. Means the formation of the gametes, the chromosome disjunction is occur. That time this type of process is occur. Here what it is means is find out the Bridges take the experiment and conduct that he crossed with the AAX, means triple X. The female is crossed with the normal male, the AAXY. Which gametes are producing means here the different types of gametes. Normally the AAXX crossed with the AAXY, but here he's taken the imbalanced progeny that is called as the AAXX, and the triple X, AA triple X, and the AAXY. The gametes, which gametes are formed means possibility of the gametes are AX, AAX, and the AAXX will be there. Maybe, and are also the AXX, AA, the next like this, they are forming R, A. These gametes are formed. Here, in this mainly the only two are formed. They are the AX and AY. Two gametes are formed. Here, the means they are crossing different types of progeny. Means these are the from the female, triploid female cross with the normal male, what will happen means this type of progeny is occur. The what it is means the see this one AAXX will be formed, AAXY will be formed, AAXO and the AAXXY and AA triplex Y and the triple A XX and the triple A XG like this. These progeny is formed the crossing with this one here. Next, just the ratio between the number of here according to the sex determination is mainly by the depending upon the ratio of the X chromosomes and the autosomes. Here, the ratio between the number of X chromosomes and the number of haploid set of the autosomes between them. The ratio is mainly depending upon this one. See this one. For example, here it is there. What will happen? The triple A XX will be formed. See what it is here where it is triple A the X here this one triple A XX will be formed. Next, like that we are adding this by the triple A XY is formed. Like that these progenies are formed by the different types of gametes they are forming, and the difference mainly the sex determination is mainly depending upon the sex index. Here what is that see this one sex index is equal to number of X chromosomes, how many X chromosomes are there and how many autosomes are there. We calculate that and we identify the organism sex. Here it is what will happen here if, for example, AAXX is there. First one we take it. In this we substitute in this formula sex index is equal to X by A, number of X chromosomes by autosomes. Clear? How many X chromosomes are there in this? 2 X chromosomes are there and the 2 autosomes are there. That's why what is the ratio? 1 will be there. Here what it is means here one thing, the 1 is there. Here for example, the 1 is there that we call as a normal female. Here 0.5 is there that is called as a normal male. 0. Here I'm writing clearly. .5 is there that is called as a normal male, and between the 0. For example, here between them 0.6 is there that we call as a intersex. Next, below the 0.3 is there. For example, below the 0.5 is there that we call as a meta male. If they are having the more than 1 that we call as a more than 1 is there, then we call it as the meta female. Here like this, depending upon the sex index, the determination will be there. We observe here, see this one, for example, we are taking the AAXX. In this, how many X chromosomes are there? X chromosomes are 2. Why the A autosomes are 2. Total, how much we calculate the 1 is there. Here, if the 1 is there, see this one here. Like this here, and also in this also. See, 1 is there. Here for example, here the 2 X chromosomes are there and the 2 autosomes are there. What it is, here it is a 1 here, and also here also you take the 2 X chromosomes are there and the 2 autosomes are there. Here also 1. That's why here the 1 is there. That's why they are the normal female and the female. Here next is next we are taking the next one. What is the ratio? We are counting. See this one. Here it is 2. The first X chromosomes are only 1 by autosomes how many? 2 are there. Here, see, I'm writing here XX, XY. Here, in the XX, XY, how many X chromosomes are there? Only one X chromosome. Autosomes, how many? 2 are there. Means the what it is the ratio 0.5. Here, like this. This is the 0.5. 0.5 is there. That is the already I said that next the 0.5 is there that we call as a 0.5 is there that we call as a normal. See this one here and here also 1/2 means 0.5. Here also 1/2 means 0.5. Here like this these are called as a normal male and the normal male. Normal male, normal females are over. Next here, you observe here, what is the ratio? What is the karyotype? The AA triple X. AA triple X. What is the ratio? How many X chromosomes? 3/2 means 1.5. Observe here. Here it is a 1.5 is there. 1.5 means more than 1.1 is there that we call as a meta female. Here like this only. Next also same. Here the Y chromosome is there, but Y chromosome not participating in the determination. Here also 3/2 is equal to 1.5. Here the 1.5 is there that we call as a meta female. Next is the triple A XY means here it is a 1 by autosomes 3 means what it is 0.67. Here you calculate it. Here the 0.6 means between the 1 and the 0.5 is there. That's why it is a intersex. Here they are not having the maleness as well as femaleness. Here next the insects are not having the maleness or femaleness. Next here it is a triple A XO. Here also same 1/3 is equal to 0.6. Here also same here they are having a 0.3. 0.3 means less than the 0.5. That's why this one is called as a meta male. And the last again the four X chromosomes are there. X chromosomes are 4 and the autosomes are 3. That is the 1.33. Here that's here also is a meta female. Like this in the who explained the genetic balance theory means Bridges are explaining the genetic balance theory. He found that in Drosophila Y chromosome doesn't play any role in the determination, and he said that the ratio between the X chromosomes and autosomes they determine the sex. Here Bridges crossed the triploid female means triploid female means they are having the 3 X chromosomes. Triploid female to normal male, the AA, XX, and form it as a these progeny is from these progeny. How we identify this one means normally AA, XY is there, we can say it as a male, AA, XX is there, female. But here different types of karyotypes are there. That's why he taken the one formula that is called as a sex index is equal to number of chromosomes by the number of set of autosomes. You calculate it like this, and what it is, one more time I explain series one. Here this one is if the 0.5 is there, that is called as a male. The sex index is coming 0.5 is there, that is a normal male. For example, it is a 1 is there, that we called as a female. The ratio means the index is 1 is there, that we call as a male. Between them 0.6, 0.7 like it is there, that is called as a intersex. Next, less than 1 is there. Less than 1 means the 0.5, more than 1, 1.3 like that. The more than 1 is there, that we call it as a meta female means the 1.3, 1.5, anything 1.3 or 1.5 is there, that we call as a meta female. The less than 0.5 means the 0.3, 0.2 like that is there, that we call as a meta male. Here less is there, that we call as a meta male. Less than 0.5 is there, that is the meta male. More than 1 is there, that is called as a meta female. Here this one is about the genetic balance theory. PDF. Oh, HHP. Our education bar. Next, we are going to discuss about the next. Next one is the in the evolution, the neo-Darwinism. Next one is the neo-Darwinism. In the genetics already that completed. The which one? The genetic balance theory we completed. Next is called as a neo-Darwinism. Here the in the evolution already we discussed about the some questions we discussed the homologous and analogous organs, and the next is the mutational theory and also natural selection we explain I explained, and the next one is called as a neo-Darwinism. Here The next, who explain this one? This means the Fisher and Seval, right? And they are explained the natural selection in the post Darwin discoveries. Clear? Next, in this mainly according to the neo-Darwinism, the mainly the that is also called as neo-Darwinism is also called as a modern synthetic theory. In this, mainly the 5 basic characters are there. What are the characters means? First one is called as a gene mutations. Inside of the gene, any changes, mutations already we discussed in the mutational theory means any changes are occur in the sudden changes occur that we call as a mutations. The gene mutations means the change in the change the structure of DNA. DNA structure is changed. That is called as a genetic mutation. Means that is also called as a point mutations. Inside of the genes, any changes occur. Change in the structure of the DNA is called as a gene mutations, are also called as a point mutations. In the DNA structure, any changes occur that is alter the phenotype and produce the different type of variations in the offsprings. That we call as a gene mutations. These are also called as gene mutations, are also called as a what it is? Point mutations. They are also called as a point mutations. Point mutations means the inside of the structure of the DNA, any changes occur that causes this type of mutations. Next, the second one is called as the second, the chromosomal mutations. Inside of the chromosomes, any difference is there. Means the addition means one chromosome is extrally added. Here that we call as a addition means the plus one. The chromosome is having the one chromosome extra that we call as addition, and the deletion means one less chromosome is there that we call as a deletion. Inversion means changing. It's changing that we call as the inversion, and the translation of the chromosomes come under the chromosomal mutations. If it is a plus one is there, any chromosome excess is there. For example, we are having the 23 pair, one extra X is there that we call as a addition, and one is the deleted that we call as a deletion. Inversion means exchanging the chromosomes that we call as a inversion, translocation. Like this, these are causing the chromosomal mutations. Clear? They alter the phenotype. Here also the phenotype is affected. In this also phenotype is affected. This is the gene mutation. This one is called as a chromosomal mutations. Next is the genetic recombinations. Clear? During the meiosis division, the formation of the gametes, that time division is occur, that we call as a meiosis division. During the meiosis exchange of the chromosome bits, and the genes takes place, produce the heritable charges, heritable variations are occur. Means the recombinant DNA is having, what will happen means the changes occur in the chromosomal the structure. That's why they are forming the heritable variations are occur. That we call as a genetic recombinations. Here when it is during the meiosis division, exchanging of the chromosomal means for example, they are, I will in the meiosis division, what happened means here mainly they are dividing. Mainly they are like this, they are dividing. It's a division time. The one extra is coming here or one less is coming. Like this, in the division time, they are causing the changes that occur as a recombinations. Here, next point is the natural selection. It favors some variations. It favorable to the some variations, means nature selection. Nature will be selected. Here, it favors some variations and reject others. Some is for some characters, it is a favor. Some characters, it is rejected. Here, next, the selected variations continue into the next generation. Nature selecting which characters are characters are continued in the generations. Which one is rejected, that is not continued in the next generation. That is about the natural Darwin's. Already we discussed in the Darwin's natural selection. Which one? Here the Bitson Batuleria. That is a pepper moth. That pepper moth is mainly They present inside in the England before the industrialization. The gray marks are more. The after the industrialization, black marks are more. Like this, the natural selection, nature selecting which one, that only the continue to the next generation. The which one is rejected, that will be disappear. Here, this is about the natural selection. Next is the reproductive isolation. Any reason which stops the interbreeding. Here, reason anyone that stops the interbreeding in the group of population is called as a reproductively isolation. Here, the interbreeding with other animals, they stop the interbreeding with other animals. That is called as a reproductive isolation. See this one. Any reason which stops the interbreeding in groups of the population is called as a reproductively, they are separated here, and the groups of the population which do not interbreed with other groups grow in the separate species. Means the, for example, here the species is there, that species is interact, they are not reproductively they undergoes interbreeding with each other, they are get separated, that we call it as a the growing as a separate group, they are, that we call as a reproductively the isolation. Here what it is, once I will again I'll explain. See this one here, the mainly the 5 characters are there in the neo-Darwinism. 5 characters he explained the in the model synthetic theory. First one is called as a gene mutations. Gene mutations. Inside of the gene, any or DNA structure, any change is there that is affected the phenotype of the offsprings. Next, second one is called as a chromosomal. Here, chromosomal mutation. In the chromosome, in the inside of the chromosome, any changes are occur. Means the chromosome number increase or the decrease or the structure is change. Means inversion, translation, translocation is there. That time what will happen means the phenotype is changed. That we call as a chromosomal mutations. Next one is third one is called as a the genetic recombinations. Meiosis division time. Here genetic recombinations. Means during the meiosis time, the chromosomal bits are exchanging. That we call it as a that time also the variations are occur. That we call as a genetic recombination. Next one is the natural selection. In the organism, in the natural selection, which one is selected, that will be continued in the next generation. Which one is rejected, that is stopped from that generation. That is called as a natural selection. Next, the fifth one is called as a the reproductive isolation. Means the reproductively they can separate. Means they can't interbreed with that group of the animals, that group of the that species animals, and they separate that way. That is formed as a new separate species that we call as a reproductive isolation. Here, this one is about the neo-Darwinism. Here, next. Next one is genetic drift. See this one. Next is the Next, last question in the evolution. This is called as a genetic drift. Here, the change in the frequency of the genes. Frequency of the genes. The change is occur in the frequency of the genes that occur means how they are changed by suddenly they are occur means not by the nature. Here, the change in the frequency of gene that occur with chance by the selection. In the small population is called a genetic drift. Here, the frequency will be changed. Suddenly, the frequency will be changed. That we call as a genetic drift. In the small population, the frequency of the genes will be changed by chance, not the selection. That we call as a not the selection. By chance, it is occur. That we call as a genetic drift. Inside of the small population, the frequency is changed. Here, change the frequency by chance is called as a genetic drift. Next, a gene with two alleles. All the genes are having the two alleles. If the frequency of the particular gene is only 1% change is there, here 1% probably of the losing that allele by chance from the small population means in only one gene by chance the changes occur that the change the frequency that is how much 1% is there that is negligible. That's why they are chance of the population. The result either loss of that allele. Here they are the end results that either formation or the loss of the allele. Clear? They are. This is about the genetic drift. Next is the genetic drifts to reduce the amount of genetic variation. Genetic drift means sudden change, sudden by chance they are occur. What that means, genetic drift tends to reduce the amount of genetic variation. They are reducing the variations. Genetic drift is reducing the variations within the population, mainly by the removing of allele with which low frequency. Which one is having the allelic frequency or genetic frequency is less, that one is deleted. Which one is more, that will be taken. Which one? Genetic drift. Clear? That tends to reduce the amount of genetic variations. By how it is within the population, removing of the alleles with the low frequency, then the genetic drift can be explained that the founder's effect. With the effect, they are exemplify the founder's effect also. Here, what about the founder's effect? Here, it is a interbreeding means the separate. Before only we discussed natural selection, the reproductively isolated from the original population that forming as a new group that we call as a founder's effect. See this one. If the small group of individuals from the population starts new colony, the isolated region, those individuals are called as a founders. For example, this one is a one group of organisms. From this, some are coming out. Some organisms are coming out. They are formed as a new colonies. Here they are formed as a new colonies that we call as a who are started that we call these are called as a founders. Here I will draw. Here this one is the original population. Actually, this one is the population. From the population, some organisms are comes out. They are called as a founders. Here they are called as a founders. From these founders, some new colony is formed. Here, like this here. The effect means small group of individuals from the population starts the new colony, and in the isolated region, those individuals are called as a founders. These are called as a founders. Next, the founders of new population, the allelic frequency of their descendants are similar. Their descendants, the allelic frequency, and these allelic frequency is the same. Here, of the founders rather than the either ancestral or parental. This is the same, the allelic frequency is there than this one. Clear? This is our, for example, see this one, presence of O positive blood group is nearly 100% in Red Indians. The forefathers of the Red Indians tribe, the predominantly positive means they are ancestral blood group only they are having, and the isolated themselves reproductively from other population. Other population they are isolated, they are reproductively isolated. That time also they are having the positive group. Here, this one is about the genetic drift. Simply, what is the genetic drift means? The frequency of the genes occur by chance, not by the selection, is called a genetic drift. This is less. The genes allelic frequency is the 1% is there, that is eliminated, means they losing the alleles chance of the population more. Here, next the genetic drift tends to reduce the genetic variations are reducing. The removing the allelic frequency less is there, that also removing from this. Clear? With this, we completed the total evolution questions. One more time, I'll explain the evolution questions. Yesterday only I explained the remaining two, the neo-Darwinism and genetic drift. Today I explained what is it in the evolution. What we discuss means the differences between the homologous and analogous organisms. Once I revise this one, homologous organisms means they are structurally same, the functionally different. Structurally same, functionally different organs called homologous organs. Which organs are structurally same and functionally different? That organs are called as a homologous organ. The reverse, the functionally same organs, functionally same and structure and origin is different. Structure and origin is different. That is called the analogous. Example, what it is? Homologous, which one? The forelimbs of vertebrates. Forelimbs of vertebrates. Next, this one is the wings of insects. Wings of insects and wings of bird. Both of them are structurally different, but the functionally same. This one is about the homologous and the analogous organs. Next, second, mutational theory. Here, the mutations means sudden changes are occur, that we call as a mutational theory. And that is called as a mutations. The mutational theory. What it is? I will show this one. Mutational theory. See this one. Once. See. This is about the mutational theory. See this one. The sudden and random changes. Here, sudden and random inheritable changes are occur in the organisms is called as a mutations. The mutational theory who proposed Hugo de Vries, he worked on which one, which plant? Oinothera lamarckiana. He found the four different forms he observed. The are called as a Oinothera brevis, Oinothera livifolia and Oinothera gigas, Oinothera nanella. Here different types are there. That and the all the forms are mutant forms. That is called as a, the Darwin called it as a force of nature. Bateson called it as a discontinuous variations. Here that is about the mutational theory. Next one is the industrial melanism. Darwin's the natural selection, industrial melanism. Here, the inside of this, in the industrial melanism, what it is mainly, the two types of the who proposed this one. See this one. Experimentally, this one is Darwin explained this one. The what it is means he observed the Bitson bacteria, and this is also called as a pepper moth. Common name of this is called as a pepper moth. In this moths, the two moths are available. They are called as a gray color and the black color. Gray color moths are abundant before the industrial melanism. Why means gray color before the industrial establishment, the barks are gray in color. That's why gray color is not visible to the predators. That's why gray color are more in this time. Next, after the establishment of the industries in England, black color is more. The barks are becomes as a black color. That's why for the predators that is not visible. That's why the black color is more in that time. Here, next, this is mainly nature will be selected. Inside of the nature, the pollution is there. Automatically, the barks are black color. That's why the dark color means black color is more. Before that, no pollution. That means which one is selecting? Nature is selecting. That's why it is called the natural selection by with the industrial melanism. Here, next this one is next one is neo-Darwinism. Now only we discuss. In this only 5 points. You remember this one mainly. What are they means? The first gene mutations, headings you put it. The chromosomal mutations, genetic recombinations, natural selection, and reproductively isolation. Here already 2 times I explained. No need to explain this one. The 5 headings are important. That 5 headings is which one? Fisher and Sewell, right? These 2 scientists are explained means that this is also called as a neo-Darwinism, is also called as a modern synthetic theory. One more name is there, that's called a modern synthetic theory. Here, next one is last one is called as a genetic drift. That means what is the genetic drift means? It is normally it is the change, the allelic frequency, the gene frequency or allelic frequency is changed with the normally it is not by the selection, it is by chance it is occurred, that we call as a genetic drift. What it is means, it is mainly the genetic drift is useful for the it is decrease the variations, genetic variations are decreased, and which frequencies are less. For example, 1% frequencies is there, that is eliminated, and which one is more frequencies are there, that is given to the next generation. And also under that only we discuss about the founder's effect. Founder's effect means from the normal population, some are reproductively isolated and there are formed as a new colony that we call as a founder's effect. That founder's effect, which individuals are there, they are the same in the descendants. Here, this one is about the genetic drift, the evolution SAQs we completed. Next to today, we discussed already in the genetics. One question we discussed that is called as a genetic balance theory. That genetic balance theory is proposed by the Bridges. Here, Bridges means sometimes normally the chromosomes are determine the sex. Sex chromosomes are determine the sex. Sometimes what will happen means the Y chromosome is not participating in the sex. Here, what we discussed, genetic balance theory. Genetic balance theory. Genetically means in the meiosis time, what will happen means the gametes are divided. One chromosome is more is there, one chromosome is less is there. What will happen means that is having the AA double triple X will be there instead of one X chromosome is extra here. So sometimes what will happen, AA XO is there means one less chromosome is there. That is occur by the genetically in the meiosis division. It is occur the genetic balance theory means it is not depending upon the karyotype, depending upon the sex ratio. That sex is mainly depending upon the sex ratio. That is by the X chromosomes by autosomes. Here, number of X chromosomes by autosomes, how much is there? That mainly determine the sex. Means, for example, one is there, that is called as the female, the 0.5 is there. Before I read 0.5 is there, that is called as a male. More than one is there, that is called as a meta female. The less than 0.5 is there, that we call as a meta male, and between them it is there, that is called as a intersex. Clear? This one is about the genetic balance theory. This one is the important question. The tabular form is also there. You practice the tabular form. The karyotypes are important. The karyotypes you get easily write it as a ratio. Here, tomorrow we will continue with the last lesson, applied biology SAQs. Tomorrow same time we will meet this one. That's it.